And we even complete. Fantastic. All right, yeah, so welcome everyone again. We have an interesting morning. You heard a very interesting talk now. And now we have the interesting questions we're going to raise here in the panel. But before we're going to do this, I would like to ask the panel members just briefly in 3 lines, basically say who you are, where you're from, and then we start with some questions.
Thank you very much. I'm Jon Atli Benediktsson, Rector and President of the University of Iceland, and my background is in electrical and computer engineering with main focus on remote sensing.
Hi everyone, I'm Greg Kerr. I'm a research scientist at the Child Mind Institute, where I lead the Center for Data Analytics, Innovation and Rigor. I did my PhD in Alan Evans' shop at the MNI. My background is biomedical engineering and high-performance computing.
Yeah, hello. My name is Luba Zeel. I'm just started at the eBrains ASPL locally, but I'm working for the Human Brain Project for a while now as a data and knowledge engineer and coordinator. I'm a biologist and neuroscientist by background.
Hi everyone, I'm Tristan Glattard. I'm associate professor in computer science at Concordia University in Montreal. My background is in distributed systems and medical image analysis, and I lead the lab on big data and neuroinformatics.
I'm Timo Dickscheid. I'm from Germany, working in Jülich with Katrin Ammons, and I'm a professor in computer science in Düsseldorf. My background is computer vision mostly. I'm doing image analysis, microscopic image analysis, and more recently neuroinformatics.
Morning, everyone. My name is Paul Thompson. I'm a professor at the University of Southern California, where I'm associate director for the Stevens Neuroimaging Institute. I'm also PI for an international consortium called Enigma that leads brain imaging and genomic studies, and you'll hear a little bit about that tomorrow morning. Thanks very much.
Right, I think you can keep the micro because we can nicely connect, I think, to Kauri. We had a nice discussion yesterday, and I thought we maybe can put it in this way. The first question where we thought about challenges going back to your career and now in integrating big data, working with data, publishing with it, and then you had to collaborate with Decode. And you had an interesting approach you mentioned yesterday to overcome this challenge. Maybe you can describe it a little bit before we pass on to the others, how that worked. I found it very fascinating.
Sure, thanks Maurice, that's a great question. So one of the areas that we work in that's possibly of interest to folks here is the connection between genomics, which we heard Dr. Stefansson talk about, and brain imaging. And of course, these are vast spheres of scientific activity. There's enormous biotech interest in genomics and the vast array of folks here who've dedicated their lives to brain imaging. I mean, they're huge disciplines. But it's rather uncommon to be expert in both of those at the same time. And I think, Maurice, this relates to a later question that you have in this session. So a big challenge that we had earlier in our work connecting imaging and genomics was essentially to try and make sure each discipline understood the other. And so if you were doing a genetic analysis of brain images, the early work was looking at specific candidate genes. And so you'll remember in textbooks about biological psychiatry, there would be a gene for aggressivity, or there'd be a gene for addiction. And this was 20 years ago greatly disproved by all of the geneticists. In fact, Dr. Stefansson was one of the pioneers in this. But you will have seen in the brain imaging literature 15 to 20 years ago studies of single genes in a brain signature. And so that whole idea that it would be very easy to find connections, I think the geneticists, as we heard this morning, revealed that it would take tens of thousands of images and tens of thousands of genomes to discover this. Not that the effects were very, very tiny. They don't have to be tiny. In fact, ApoE, as you saw, has a pretty major effect both on life and on the brain. But because when you are searching the entire genome, the risk of finding something by accident, which of course people had been doing, had to be mitigated by having replication on replication. And so I think, just to answer your question, early on, and Marianne's talk yesterday addressed this very clearly, it was very uncommon to share either images or even more so genetic data. And so the way that we addressed it, I'm not so sure that this is the best way, but it's one way, is in work with Dr. Stefansson's team at Decode. They would run exactly the same analysis on their images and genomes as we did, and Enigma, as you'll hear about tomorrow, was repeating this in different countries. And at the very end, you would say what you'd discovered. And I remember it was very ginger in those days. People would say, well, we'll share our result with you if you send your result to us first. I mean, can you imagine how evasive and non-cooperative this was? But I think it was such a different philosophy maybe 20 years ago about sharing discoveries and sharing results. So I think even today, studies that use genetic data, they tend to keep the genetic data where it was collected because of privacy reasons. And so this solution of not so much shipping data around, which is valuable in many contexts, I think it was slightly less common and certainly less popular among geneticists because of those reasons. But it ended up in a very successful collaboration where, in fact, the genomic loci that were discovered in Iceland as of influencing brain volume, influencing brain function, you saw some of these this morning, were exactly the same as the genetic loci discovered in North America, in Western Europe, in Australasia. And so I think that gave people confidence that these results went beyond the candidate gene studies that didn't replicate into something that was much more secure and replicable.
All right. No, I think that was a very interesting approach to data silos, but still having an interesting publication out of it. Marianne, you were also part of the panel. I realize, I'm so sorry, I have to apologize. No, please come on. We just have a seat here for you. We were wondering why there's an empty seat. So
you have to answer.
I'm terrible sorry. I tried to get Kauri still here on the panel, so was a bit distracted. Maybe shortly introduce your name as well.
Yeah, so I'm Marianne Martone. I'm a professor of neuroscience at UCSD. I started my career in neuroanatomy, but the last, I'd say 20 years, 30 years, 25 years at least, I've been in neuroinformatics.
Right, okay, and you connected us already in your keynote a little bit about the biggest challenges that you said.
Yes.
Maybe you can elaborate a bit, going back to all your career, what have been the biggest challenges with big data and all these projects, and how we basically can overcome this, perhaps?
Yes, so I started my career, as I said, in neuroanatomy, and I did my postdoc with Mark Ellisman at UCSD, who I think was one of the first who was really trying to bring computation to biological data. So this was in the area of microscopy, really pushing digital imaging. I always tell the story that I was recruited to his laboratory because in the early days of 3D reconstruction in my graduate work, you used to have to set a camera up in front of the computer and take a picture of a 3D reconstruction. And when he was trying to recruit me to be a postdoc in his new center, he goes, and you're going to just be able to push a button, right? And the camera's going to communicate with the printer, and you're going to be able to just print a picture. And I thought that was the coolest thing I ever heard. I'm like, wow, right? The future's here. And it was this little tiny picture about this big. The color printers couldn't figure out how to do blues at the time. Blues were very difficult, and so it looked very distorted. But still, you kind of recognized that we were on this trajectory. And early on in particular, as you saw the growth of computing, we were associated with the San Diego Supercomputer Center, and the size of images that you could reasonably process were very small. I mean, very, very small. As we know, compute power really went up. But we saw very early on that our capacity to generate data of a certain size, being able to mosaic tiles together, being able to push dynamic range, vastly exceeded our ability to actually do anything with that data. So almost immediately, we would take these gigantic images. They would be beautiful. You would look at them on a tiny little screen, first of all, so you had to constantly be zooming in and out, so you really couldn't appreciate it. I do remember when the first tiled display came in. This was sort of an early version of the BigBrain, and finally, you could see one of these mosaics at full resolution, and how much detail you could see when you got to see both detail and context was really amazing. But in this, I think, Paul, you alluded to this, in our world of informatics, and I think, Libya, you'll probably talk about this too, it's, if we're going to make this data public, what are we going to make public? And is it going to be this full resolution thing that nobody can use anything with that taxes our storage systems at the time? Or are we going to immediately downsample and make something that is tractable because we can then reasonably handle it? And over the course of my career, these trade-offs have always been there. And there's this sort of sense, I was just on a panel, I think last week, 2 weeks ago, where again, people think that we can infinitely scale our data sizes and that the infrastructure will be there to handle it. But not when you're looking for stability, not when you're doing it at scale across everything. Every time there's a new format or some new technology that comes in, research scientists assume, right, that you should just be able to hand this over and that people are going to be able to make your data available, and they're not going to be able to do that. That's not going to happen. So I was on a panel at the National Academy which was looking at long-term costs of biomedical data, and we heard from people at CERN and everything else, and even at CERN, they throw away most of the data before it ends up being put public. So I think from an informatics perspective, this is a tension that is always there.
I think that's a good point, and leads me also to remote sensing. Even if we are here in the neurosciences, I think the rector here, Jon Atle, you have experience way back when we had very small data sets. We're talking now about hyperspectral, multispectral data sets. So it's actually also large data sets. What do you think were the challenges in this domain? And then when we look back on the neurosciences, what are commonly points where you think we can master these challenges with the same approaches, right? For instance, in the supercomputing centers, what can we do to help multiple domains?
Thank you very much, Morris. Yes, I come from a different field, but there are many similarities. Way back, you mentioned, I was doing this 35 years ago, and then big data sets are small now. I mean, if we look at it, what was happening 35 years ago, I started doing classification. I mean, it's basically information extraction from data sets. So I had data from multiple data sources, and that's what we like to work on these days with big data. For the first stumbling block was that in my lab in the US at Purdue University, we very much focused on statistical approaches. That was the way forward. But it was very difficult to model when you have data from multiple data sources. So I went into computational intelligence using neural networks and later random forests and so forth when things moved on. And that was successful. And then I started combining things, and then we had hyperspectral data. And those were big data in those days, 100 megabytes. I mean, we had to find a way to process them. So we found that using some assumptions in the fields and so forth. But basically, moving forward, the data becomes bigger and bigger. The algorithms become better and better. And in the 80s, we were in the second wave of the neural networks. And it was so funny. I mean, you started the algorithm and it ran 4 hours and you looked at it the next day. I mean, it's the same thing happening today, but you just have much better algorithms, much better computational power, and much bigger data. So we are essentially working on the same field. We need more training samples to make accurate assumptions. And what was the most challenging thing we had in my lab, I'm now only looking 10 years back or something, was when you were not only looking at the spectral information, we were looking at the images, spatial information, to combine those. And then we looked at pre-processing, and it's basically feature extraction. What are the best feature extraction algorithms? And it's always the same. It's you need more labeled data, you need better prepared data. You don't want to throw away data because, I mean, there can be interesting information for a scientist there, but there's always the trade-off between the known samples, and then you need the human capabilities to label them, and the algorithms and the processing. So basically it's the same, but I mean, it's always challenging, but we want to create better approaches and get better results.
All right, thank you. I think you put on a very interesting point. I remember Gabriel here in the room, right? Mathematical morphology, we had a PhD together there, and really deep learning was not there. So suddenly deep learning came with convolution, take neighboring pixels into account, a different approach, a new approach. And when I think about Timo, you work with AI, but you also have a quite interesting background in computer vision. So the traditional methods using, rather other elements of have few neighboring pixels involved. What your challenges there in terms of data with the, traditional methods you maybe have, and when you vice versa this with all the new deep learning approaches you have?
Yeah, I think there has been a big shift now with large data sets, because when I did my PhD, it was still like, you focused on a project, you got your package of data that you wanted to work with, and usually, you had that on a disk, you took it with you for the time of the PhD in your office, and that was the stuff you were working on. And nowadays, it's we have these very, very large data sets, which nobody carries around. They sit, in our case, very often in a supercomputing center. And you have these algorithms which run in parallel and do things, like deep learning. Let's say in our case, typically would be things like segmenting cells or something like that. And you run that on a large scale, on a lot of data, because suddenly there is this algorithm which actually can give you numbers, which gives you good results. But then there is also this huge pace of developing new methods. Even the same model can suddenly become much better as you find a new way of, let's say, with self-supervised learning, training it. So suddenly there can be a way that your model becomes much better, or there comes a new model. And it might be that you get much better results out of what you just did with a lot of computation, and you would like to do it again, very, very quickly. And I think this is, for myself, I see that this is really a big change. That was not something we faced previously. You want to have everything ready, set up with the infrastructure, so that you can rather quickly recompute, redo analysis at a large scale, and that's of course expensive, but you don't want to miss it.
All right, yeah, very good. You mentioned high-performance computing capabilities, and you mentioned that as well in your introduction. When you're using these machines, what were your challenges in your career so far, and what you would expect when we think about the big brain evolving to higher resolutions and so on?
Yeah, and I mean, I guess to also tie this back to an earlier point that was mentioned, I think a big key challenge is one of doing feature extraction. And I think this is ultimately a bottleneck that impacts everything else we're doing. Because if we don't have good feature extraction at an early enough stage, then we will continue to hit these bottlenecks of we don't have enough storage, we don't have enough compute power, and these sorts of things. We'll be working on massive dimensional data. We won't have the resources to scale to it, or we'll collect this next generation of image. By the time we have done that, all of a sudden now the current state of computers can't keep up. And so I think that this piece is a crucial one. And so figuring out how we can do feature extraction in a meaningful way, but then also, again, using platforms, whether it's tools like Brain Viewer or things like this that allow you to actually verify that the features you're getting are meaningful. And I think so in the high-performance computing space, that's been probably the largest challenge I've encountered, is that we have massive amounts of data. For this grant I'm working on currently, we're processing the extent of the ABCD data sets, or around 10,000 brains, each with 200 fMRI pre-processing pipelines. And so these are all permutations of commonly used pipelines that are widely adopted in the field. And the goal is to study analytic variability and understand how these things are different. But before we want to draw any insights from the space, we need to verify when the pipeline did and didn't crash. And as often happens in these spaces, you'll produce an output file one way or another. But how you can actually understand that these are the meaningful features we're getting at the end of the day versus these are meaningless ones. So you can do this filtering so that you can then actually go on to perform meaningful analyses downstream. I think this is a big challenge that has yet to be solved in a way where we can be kind of semi-human in the loop, where because it's going to be intractable. You can't have hundreds of thousands of images being manually rated by an undergrad RA. It's just not tractable.
Mm-hmm. Yeah, thank you very much. I think you worked on a couple of pieces like pipelines. I think you have these tools mentioned that we want to integrate in something called CBrain. I look at Tristan, right? We work on this together a little bit. And automation, because experts like you working on this daily, 10,000 brain data. Maybe Tristan, when you think about the challenges, what we do with CBrain to kind of get to this, so how we can semi-automate or something like this complexity that we have in these brains and in the brain data sets. Can you?
So maybe one thing, that I think is a particular challenge is how to work openly with these large databases and experiment at the scale of what Greg was mentioning. So I feel like in the past decade or so, as Paul, Marianne have mentioned, like we've become quite good at sharing large collections of data, but there's still, I think, an important challenge in documenting, working openly, having, sharing analysis of these large databases. So maybe to relate to that, I'm PI of a project called Living Park, where our goal is to sample the Parkinson's imaging literature and redo some of the analysis and share these analyses publicly in the form of Jupyter notebooks. And I think it's both, interesting but also difficult. And maybe two big pieces. First off, when we talk about open data sets, they are in general in our field not 100% open, in the sense that you still need data usage agreement, you still need authentication, you still need various forms of technical and non-technical restrictions, which are very legitimate. And so it's not that easy to share code, share analysis that are going to handle that. In particular, if you cannot share intermediate results, if essentially you have to make sure that people can recompute these results, which is not trivial. And the other piece is increasingly we rely on important infrastructures. You mentioned CBrain. I think it's getting very difficult to run any decent analysis without a high-performance computing cluster. And these infrastructures are not public by definition, and they won't be. They can be shared among a community in Canada, in our case, in Europe, but you still need to have accounts and restrictions for security reasons. And I think sharing analysis in this context, when they require to run on these platforms, is another challenge.
Okay. Thank you very much, and I think you connect nicely to Lübje, because we also yesterday discussed with Ron, how do I know where is the data? How do I know which scientist is working on which data? How is it can be described well? And I have been in your hands-on session, I think, with metadata. There are lots of challenges along the way, right? We have yesterday this fair, you need lots of time to do it. There are lots of, let's say, ontologies for each domains to share data in metadata. Can you maybe explain, with your background in metadata, what were the biggest challenges so far, and maybe how we can address them, especially when we think about the big brains again?
So I think my answer would be, there are two sides of the story. So on the one hand, there need to be people in place to actually do this preparation. So there was beforehand the mentioning of people have to prepare the data for sharing and they have to label them. And of course that is time consuming in itself, and it also needs a certain expertise. Marianne was mentioning this yesterday very explicitly, that it's not enough to just put a master's student on it and say, well, do the data management for us. That's typically not going to work. They lack the experience. I mean, except you're very, very lucky and have a very, very talented student. But that's one side of the story. The other one is systems, infrastructures, but also analysis pipelines and so on. They require a certain set of information being in place so that data can immediately be used. This is true for CBrains, this is true for pipelines in eBrains, this is true for the Zebra toolbox. And these kind of information, of course, not all researchers have them in mind. And for this, of course, standards are developed, but standards are typically very focusedly developed for one purpose only. And connecting those, making them available, make these different types of modalities speak to each other, right? So coming from the pure scientists that just want to put the authors on to their data sets, who created them, maybe tag them with certain keywords, but then there are deeper information that need to be in place in order to make those data reusable or applicable to certain pipelines, or to be able to be loaded into certain infrastructures to be computed on. And these kind of information the normal scientist doesn't have directly in mind when sharing the data. And there are also different approaches to this from the technical point of view. So these standards should describe the neuroscience or the information on the data themselves, but they should be also working with the technological aspects behind those, so like the tools and the systems that need to talk to them. So there are the technical parts on it and the semantic annotations. And for me, that was a big challenge, like talking to the people from the different systems, trying to figure out their constraints and requirements, and then trying to fit that all together into a standard.
All right, thank you very much. Yeah, I also encounter lots of problems. I think I remember assigning to a buoy that had time measurement data a DOI that is constantly going in data. How you do this, right? There are lots of technical questions. We should do it fair and give it a DOI, but it's keep changing all the time. So how you make accurate statistics with metadata on that. There's lots of challenges on the way. But I would like to open a short round if anyone has any burning question right now to the panelists. Right now, going once. I hope we had some coffee outside. Yeah? Okay, going twice.
Thank you. So my question concerns the problem of n squared, connectomics. It's all very well if we're getting bigger and bigger data sets and we want to process this part of the brain, that it's a voxel or a region or whatever. You can come up with coarse-grained, massively parallel algorithms to speed things up. But when it comes to n-squared problems, connectomics, you can't do that massively parallel. The results in this part of the brain are contingent upon what's happening in that part of the brain. So how do you deal with n-squared problems as n gets larger and larger and larger with for instance, the big brain is now small. It's only a terabyte. If we go to 1 micron isotropic, that's going to be 8 petabytes. And we're not going to solve that anytime soon. Now try and consider doing connectomics with data set that's 8 petabytes. You don't have the architecture to handle that just in a local problem. But what can we do to deal with the n-squared problem, the connectomics problem? Are there any architectural insights that can help us here?
Yeah, thank you very much, Alan. Yeah, please, Timo.
I'm happy to say something about it. Maybe I should not be the only one. So we are fully running into these problems, of course, at least on my side. This is the case. And there are just two strategies, which the one is to approximate, of course. Yeah, that's what people do. So if you have long-range interactions in your data and want to do optimal computations, you approximate in some way. That's what, people do in Markov random field theory, for example, that you somehow try to approximate these things so that becomes somewhat tractable, and you still believe you're close to what you want to compute. And the other thing is that you hope for the developments in computing, right? We are all hoping for exascale, and luckily enough, the HPC people are hoping for us to bring the proper problems to exascale. So I think it's these two things, yeah, approximation and working closely together with the infrastructure. That would be my.
Yeah,
Gregory? Please.
Yeah, I totally agree. And I think on the topic of approximation, this is something that's been familiar for a long time. The idea of neuroanatomical atlases is truly one of approximation. Rather than looking at every voxel, what we're trying to do is group them, and then we have fewer connections that we need to be monitoring. And so, I think it's also about taking an iterative approach. So we can start with, a coarse-grained parcellation of some kind, where we're grouping things, we're making approximations. From this, we can generate a hypothesis of maybe where do we need to look more closely. Which pieces can we, tractably evaluate, and can we restrict our question to only address these elements? And so I think that there's a necessary compromise where we can't answer all the questions all the time. And so I do think that this iterative, kind of hierarchical approach is something that needs to be increasingly deployed, so that we can also make sure we aren't just, throwing a bunch of fishing lines in the water and seeing what comes out, but we're actually doing targeted analysis. The age of blanket analysis might be done.
So I have nothing on the technical side, but I will say one thing that does seem to be happening more now compared to even a decade ago when we had people from astronomy come in and people realized that there was a lot in common between neuroimaging and astronomy. And anesthesiologists got up in one of the early brain planning meetings in the US saying, you know, we're still using signal processing algorithms from the 1950s. We have a feeling that things have moved on, but there's been no cross-fertilization. And I'm starting to see a lot more of that. I'm starting to see a lot more things that were developed in one domain start to translate much more quickly. The XAR imaging format is one of them, which we're now getting people in biology saying, hey, we're going to use the XAR format, which it turned out was developed in the geospatial community, I think. So I do think that is one of the benefits now of having shared data sets, is that the people who are developing these algorithms can also now start to apply them across domains. And that was one of the benefits, I think, that does start to flow back from making these large data sets available.
Tristan, you want to add something?
Maybe just a quick note on infrastructure. So for the kind of problems that Alan was mentioning, I feel like 10, 15, 20 years ago, we, like academia and public infrastructures, had an edge in terms of computing power. So we can think of EGI in Europe and Exceed in the US and other supercomputer projects. I feel like now the balance is shifting a bit towards industry, which is good, and it's nice that industry also has some computing capability. But I'm also fearing a bit that we may, well, academia and public infrastructure may be a bit lagging behind now. If we look in AI, for instance, it's very difficult for academic researchers to do any meaningful work on large models, if you don't have co-affiliations with one of the big ones. And so I think public investment in national computing infrastructures is really key to sustain our ability to tackle these problems.
Right. Yeah, please.
I think Alan's question on these n-squared intractable problems, Marion, your comment made me think of borrowing methods from other fields. And so I got to tell you a story. I used to ride the elevator every day in Los Angeles with a scientist who did machine translation. So he was translating English into other languages, and I'd say, how's it going? And every day he'd say, it's not going very well at all. Years later, I read a paper with him as first author, and I think it might be the most highly cited paper in deep learning, called Attention is All You Need. Attention is all you need. This guy, he's called Ashish Vaswani, who's very, very famous for inventing the transformer architecture, which is at the core of ChatGPT and other things. If you go to MICI next week, which is a medical imaging meeting, everyone's using vision transformers. So if you trace the lineage of that trajectory of approaches to the problem, Ashish Vaswani, when I would talk to him about translating language, he said, we've compiled the biggest dictionaries of Korean, English, Russian, and it's still not working. We can't think of a way of pairing up the words in one language with the words in another language. And of course, the sort of gestalt shift that, if you read the attention papers, comes into play, is that there isn't a one-to-one mapping. And when you're at a certain word, it's dynamic what the attention should be looking at. And so I think, to answer your question, Alan, the attention problem, if you look into the mathematics of it, it is an n-squared problem, or maybe worse, because you're in a language situation where you have to consult all the prior words. And I think a very elegant mathematical approach to that, which is transformers and attention, now seems to be making its way into disciplines like our own, looking at medical imaging or possibly remote sensing and other areas. So I think one answer to your question might be that this, and Marion, you said this, the borrowing of mathematical and scientific insights from other fields. And you and Katrin have brought us all together to think about these things. It gives us a leg up on the ladder, so to speak, even when the hardware can't possibly be increased. I mean, it's that sort of thing.
I would say that's a very good statement, and I have to confirm we in remote sensing we use the transformer architecture, but it brings me to a little bit a point which is sort of the next question. I would start with Jon Atle, the scalability concern here, right? We had experts, and this scalability is a term can be interpreted in many ways. Resources, human resources, experts in this field. We have talked about computing capacity. EuroHPC is there, big machines are in Europe at least available. But where do we have the experts for this? Scalability of data sets for the integration. So when we have this all as a multidimensional problem, so to speak, where do you see we should most emphasize on? And in particular, I think as rector, you have the strategic overview of the whole university, and you see, let's say, all the different domains, you know, evolving in different directions. Where do you think with all of this, more data, more compute, more skill sets, people that actually do the work and not potter off to industry maybe, and being in science? So where do you think we can put an emphasis on, and where maybe University of Iceland also goes certain ways already?
Thank you very much, Morris. For our community, if we start there, we have a nation of 400,000 people. We're very small. So in terms of the scalability, we need outside help, for sure. We need more manpower, so international collaboration is vital. We can say, listening to Kári this morning, I mean, we have lots of important data, for example. We would like to have them more available. We know there are restrictions, and obviously Decode works with University of Iceland and the hospital and internationally, and we have the UK Biobank that they use. I think having more people work on our data helps us greatly, but we also need more people to be active here in the community. So I think that's the most important thing. And one thing I have mentioned talking to politicians here in this small community is that we have, like Decode have pointed out, we have data sets here in this society that we could make available for everyone and make Iceland a test bed, a laboratory, because we have also very good mapping of our people and so forth. So I think collaboration is the key. Obviously, like you mentioned, the scalability in terms of computational activities is very important, and there we work with you and Júlich, and going forward we have very strong international collaboration networks. We need them, but I mean, I think going forward there are so many venues we can take, and for our small community, having outside help really helps, and also working with industry. It's really a key thing.
All right. Yeah, thank you very much. Talking about when we think about satellite imaging, we did an exercise, me and Gabriel, where are you, here somewhere? And did the metadata on satellite images. We did, you know, all the runs on HPC, putting metadata on this, collecting this in a repository. And it was huge, humongous time that actually went into this. We had a nice journal in JSTARS, it's a remote sensing. We got really good appreciation for it doing so, but it was a lot of time. So in this regard, I think given the challenges of more data, more computational systems, we talk about AMD GPUs, NVIDIA GPUs, Intel GPUs, you have to also describe how the data was maybe generated or analyzed. When you see all this complexity coming, and right now we don't even have the chance basically of doing it properly, I guess you would agree we do a terrible job, right? It's just so much work to do the metadata all the time. What would you think would be the future? More skill set building on this? Should that be more put in grants as a necessity? What would you think about the challenges having more large quantities of data ahead?
So there are also different aspects to this. So what I see is that generally in education, the way of learning how to describe data properly for others is not really taught at the moment widely. So that's one big lack we currently have, and that we have to tackle from the university side to actually make this a topic, so that researchers learn early on how to deal with this. There are certain principles that they could learn very early on, and that helps a lot. Also, the way of working in parallel, so what happens at the moment in a lot of cases is people collect their data, and as soon as they have to share, I think Marianne showed this yesterday as well, as soon as they have to share it, then they start about thinking about the metadata and how to describe data and prepare it. And I tell you from my own experience doing this in a collaborative work with people that generated the data elsewhere, and then I had to publish those data and work back through the metadata. This is an enormous work if you have to do it for the, like, when everything was already done. Then it's kind of, accused of being an investigator, an interrogator, when I was then talking to the researchers, because I had a lot of questions, and he felt like being in the spotlight and being accused of not doing his job. And he did, right? Everything was there, but it was distributed all over the place. So starting to work differently, starting to collect metadata in parallel with the data, already thinking about this is another way of making this more scalable and reducing the work along with it. And then there were other things mentioned, like tools, of course, like ChatGPT. They will have a future in helping us annotating data in parallel. So personally, I don't think there is a way around trying to do or solve these information that we need to gather also from the programmatic point of view. And there's also then the responsibilities of the tool makers, right, to incorporate that already into the tools, so that metadata or data that are produced by certain scanners, for example, that they actually have all the metadata already in place. Also that workflows that are developed, that they produce the metadata alongside and that they're stored with the data directly without a person having to collect those on the way. And then lastly, I would say, you mentioned if there should be an incentive of dealing with this from the funders, from the grant perspective. And my experience so far is that every grant or every project that is granted is supposed to deliver this, but actually giving money to funding, from the funding, so really dedicating money to somebody who is taking care of the metadata management is typically cut out of the funding very, very early on. It's just expected that you do this with master students or PhDs alongside, and that's a wrong assumption. I think it should be really explicitly money for this, so that people like the researchers are actually supported with this along the way.
Mm-hmm. Yeah, please extend.
So I think that it's absolutely true that the way that researchers think about data in the biomedical culture is, I think I mentioned yesterday, as an appendage to the paper. It's not a primary product of research. And I had an NIH program officer talk to me. I think her husband was in NOAA or NASA or one of the big, the big agencies. And she basically said that data is in their blood, and they understand that their job is to generate large data sets for people to consume. They dedicate 20–30% of their budget to this, and they have the people to do this. She said, at NIH, there is no culture there for this, right? You give out money for research, you generate your research, you publish your papers, because for hundreds of years, thousands of years, that was all we could do, was communicate about what we saw, right? The collections, the physical data was someplace else, and that's just simply what it was. So our entire system grew up without dealing with the data, except in these abstractions and papers. When I first started running databases and curating, I thought it was really interesting when scientists would put their data in, okay? If they were to put a figure in a paper, they would describe it. We would say, please put a figure with your data set, and the description would be TIFF image. That's what they would put, because they're like, this is data, I guess you need to know that it's a TIFF image. I'm like, no, it says that in the extension, we don't need that. What we need to know is what this is about. And it's why I show that example in the brain, one of the brain databases, that when people put their data in, you would never publish an abstract with a title that's called AB57 and then no description. But it just doesn't occur to them to do this. It is not the way they view data. And I think it's going to take a while to make that shift to, it's the data that you generate that we want to know. Your insights are great, but I love the fact that you're running the ABC on all these other pipelines, because what we are finding out is if you put that same data set out there, you can get a whole lot of different insights. And we have not yet tackled the influence. We see distressing things like it matters what operating system you are using and all kinds of things that will influence what you end up getting out of it. So I do think that there's some training that does need to go on in graduate level. You learn how to write a paper, you learn how to write a data descriptor. But I ultimately believe that it's the instruments, a smart laboratory, whatever you want to call it, is going to have to capture that. there is a distressing thing that happens, for example, in microscopy, where if you have a proprietary format, you have beautiful metadata. If you translate it into an open format, the metadata gets stripped out and lost. So I think that this problem has to be tackled again on a broad level, and that we're not going to see that shift immediately, but hopefully it will start to happen. But it was, it's really been interesting to see what it is, the way that people, researchers think about data. That's the thing you throw away, and it's the insights that you keep, and we're trying to flip that.
I think you want to extend, Timo, or?
Yeah, just a little thing that, because I agree very much with what Marianne was saying. I mean, we work with a lot of people in the Human Brain Project who do data, and then we help integrating it. Luba does a lot of that work. And what I also observe is that there's still a very wide impression that the metadata curation is something you do at the end, a little bit like you clean up the workshop after doing the work, then when the work is done, then you do that. But the problem is that it's very often too late, and then it is also not a nice work anymore, and sometimes not even feasible. And it has to become part of the way people work. And I find that really a challenge. It's easier to tell people at the end, please take some time with us to do it, than telling people change the way you plan the whole thing. Yeah, so you have to do that from the beginning, and each day, so to say, make that a habit of doing that. And I think there's still a long way to go to also provide the tools and the teaching and all that to do it.
Maybe Tristan, you can extend. I remember, doing lots of these things, and there was Blue Gene area, and we talked reproducibility. We did everything on the Blue Gene architecture, job scripts, and suddenly Blue Gene was stopped by IBM. So much for reproducibility, right? When you think about our toolings we create, some things you mentioned is automation automatically by tooling. Can we do this? Can we?
I don't know about that, but I'm going to follow up on what Marianne and Timo were saying before, if you don't mind. So I 100% agree that describing the complexities required through some form of metadata standards and their evolution. I also think it's important to think about how to robustify the data analysis to all these small things. So yes, operating systems, but also CPU, GPU models, compiler versions. I mean, these things have evolved a lot in the past years. Not to mention different analysis pipelines, and we can go up the stack, different scanner acquisition devices and batch effects and that type of things. And so yes, so when I think of scalability, I also think in terms of viability and complexity of how people acquire and process data. And I think it's important to also think of methods and strategies on how to aggregate data that may not have been acquired or processed in the same conditions, because we can't possibly know, how many threads were used on that particular CPU of that particular, cluster that was only used for part of the analysis. At some point, it's difficult to describe all that at a finer granularity. So let's try to be robust to that and this variability. I think that's an important component for scalability.
Right. Okay. Yeah. Gregory, please.
Yeah, and one thing, I really loved your analogy of it's like cleaning up the workshop after you've done the work. Because I think what we're entering into is also a space where there's a higher standard that needs to be met in terms of our own accountability when we're describing and sharing our data sets. We're talking about this differences across analysis pipelines, numerical states, compiler versions, what have you. Well, just because in my analysis I may not have deemed all of those details important, doesn't mean that they shouldn't be recorded and communicated. Or just because, I found that pipeline A was the only one I got a significant result on for my hypothesis, therefore it's the only one I'm publishing. Well, that's of course deeply flawed for many reasons. And so I think that we also, again, have to have a bit of a culture shift scientifically towards being comfortable operating in this, as automated as it can be, documented space where we're sharing data in a fair manner, so it can be, interoperated. It can be accessibly understood what actually took place in the generation of this data, and that can also be, presented in a way that it's not just exhaustive, because then it no longer becomes accessible, but what we also need is some structure therein. So you can, depending on the types of questions you want to ask, depending on the ways you want to interpret the data, you can efficiently understand these attributes and use them. So I think that this is really a culture shift where tools will have to lead the way in doing the bulk of it, but we also need to just societally make, come to some sort of consensus, or at least agree on a few possible consensi for how we can incorporate these things into science moving forward, and incorporate, even how to document or describe them in papers. Because, it's common, you'll have a results section with 4 figures. Well, what if you repeated your analysis under 20 different conditions and situations? All of a sudden, that's an overwhelming amount of information. So even thinking of how do we revisit the entire form of the way we're publishing results to effectively communicate and manage this kind of deluge of information.
I wonder sometimes, Paul, you're the final speaker now, sorry to put you on this, but I love the elevator and the attention mechanism in talking to someone. When you think about pipelines, now it's very technical. How you as a neuroscience would say this would be an elevator of talking to someone else, developing a pipeline where you can publish with in ways further in the future of having new ideas and on. Do you think there's some potential in this?
Yeah, I think you're right, Maurice. I mean, yesterday was a great example. So people who develop tools, I had read their papers, and I'll be the first to admit I didn't either fully understand it or get to the end or have a full. But having a hands-on workshop like we were at yesterday, a huge advance for me is the availability of Jupyter notebooks. And so you could run, and it comes back, Greg, to your analysis, you could run a particular form of analysis and see the result. You could then change the code, as was being demonstrated yesterday, and see if that influences things in a material way. I think if you could publish those workflows as well, and maybe it somewhat addresses, Greg, your point, you could have the paper, and then you could have the ability for an analyst who's interested to jump back into the data. And I think, given how good these tools are now, I mean, they're at quite a high level. I mean, if you don't have to be a computer scientist to follow the logic and the steps. I mean, maybe to write the code you do, but to follow the analytic logic, you don't. So maybe that goes some way towards addressing this and sort of democratizing the workflows, but also at the same time addressing, Greg, your point about reproducibility. What if we did something else? How would that have made a difference?
All right, thank you. The pleasure of the lady to finish. Lipia, you wanted to add something, but then in the light of the time, I think the people are ready for coffee.
I will end with a very, very small advertisement, because the topic just raised. I want to mention that within eBrains we have the concept of live papers that can accompany research papers. That means that, for example, a workflow that is published, or a model with certain validation tools that is published in a research paper, can actually also be published in a live paper where everything, the code, is published as well, and then the user can tweak those, run different parameters, see what the result is, or can repeat the results that were published in a paper. And I think these kind of concepts, despite the fact that I'm now making advertisement for eBrains and for the live papers, but these kind of concepts are very nice. And I think future publication will go in this direction, that we are less rigid and more like interactive with what we are actually publishing in research.
Okay, I think in the light of the time, I'm sure some of you have maybe still some questions, but the people will be around, I think, outside in the coffee, so please help me and say thank you once again for being here on the panel. Thank you very much.